In this video, I will talk about the latest additions, the reproduction of cells in NeuroCellSim, and how to generate new neural networks in the process. In this new version, as anticipated in the previous video, the ability to view the graphical representation of the neural network as if it is a spiky neural network has been incorporated, even if the option to function as such is disabled. In this way, each time the electrical potential of a neuron is filled, the outgoing connections will flash. On the other hand, as a new feature, the ability to view the neural matrix along with its graphical representation has also been incorporated into the information panel. In the left column, all neurons are represented in the context of departure neurons, and in the upper row in that of incoming neurons. Certain global statistics have also been added based on information from the world and from the set of cells and neural networks. This data measures the number of living cells, the amount of food available at this time, the average number of hidden neurons per cell, the average percentage of connections used among all possible, and the average variance of the weights. When regulating neural networks mutations during reproduction, NeuroCellSim uses 3 specific and 1 general mutation parameters. These parameters can be interpreted as probabilities or rates of mutation. Rate of weight mutation. This parameter defines the probability that each connection has to change the value of its weight for a random one. It controls the ease or resistance of the weights of existing connections to change. Synaptic mutation rate. This value represents the probability of changing the state of each entry in the neural matrix capable of hosting a connection. That is, some existing connections might disappear and new connections might appear that were not there before. Neuronal mutation rate. This mutation rate is associated with the probability a cell has of gaining a new neuron with random connections or losing an existing neuron along with its connections. Overall mutation rate. This general parameter works as the rate of mutation of the other probabilities. Thus, the previous 3 rates of mutation are variable over different generations and tend to diverge over time. In NeuroCellSim, there are 2 types of reproduction, sexual or asexual. For asexual reproduction to occur, it is only necessary that the reproductive cell has enough energy for itself and for the new cell. The way NeuroCellSim generates a mutated neural network from a previous neural network is as follows. It starts with an exact copy of the parameters and neuronal matrix of the progenitor cell and goes through 2 processes, a DNA mutation and a network mutation. In DNA mutation, the new cell goes through a process in which some of its inherited parameters and the mutation rates themselves are subjected to the probability of changing. In network mutation, it is the neuronal matrix that undergoes a mutation process in the 3 degrees of freedom in which it can be modified. That is, weights can be modified, connections can be created or destroyed, and neurons can be created or destroyed according to their respective probabilities. All these changes can manifest themselves in the new cell with a different color if they are sufficiently noticeable. At this version, the option of sexual reproduction has also been implemented. For sexual reproduction to occur, 2 requirements must be met. Both cells must exceed a certain energy threshold, slightly lower than in the asexual case, and they must be close enough, within the ranges of vision. The process of sexual reproduction is more complex than in asexual reproduction due to the different types of neuronal matrices that can be combined. The main challenge is represented by the replication of cells with neuronal arrays of different size, that is, with different numbers of hidden neurons, but let's start with the simplest. In NeuroCellSim, sexual reproduction also begins with an exact copy of the parameters and neuronal matrix of the cell with the largest number of hidden neurons, if they have different sizes. The future number of hidden neurons of the new cell is randomly selected from the range of the parent's numbers of hidden neurons. Then, if the progenitor cells have different numbers of hidden neurons, the size of the new neuronal matrix is adjusted according to its number of hidden neurons. If the parents and the new cell have the same number of hidden neurons, each entry is simply chosen with the same probability between the weights of the parent neuronal matrices. If the parents had different numbers, it would be necessary to differentiate between the following situations. The new cell could have the same number of neurons as the smallest of its parents, the largest or an intermediate number. For example, if the new cell has a larger neuronal matrix than one of its parents, part of its neuronal matrix will be a mixture of both, while another part will depend solely on the parent with the larger neuronal matrix. After this, the new cell goes through the same DNA and neuronal mutation processes as in asexual reproduction. In the following video, the final version of NeuroCellSim will be presented, as well as some comments on future plans and possibilities ahead.